Bob the B cell, can we bind it? Bob the B cell, yes we can! Binding infection or foreign bodies starts production of antibodies. Antibodies bind to antigens, working together to immobilize them. Bob the B cell, can we bind it? Bob the B cell, yes we can! When foreign cells are bound up tight, they can be killed by the immune system. Bob the B cell, can we bind it? Bob the B cell, yes we can!
Hello there, Bob. What are you doing?
I'm producing antibodies at an astonishing rate of 5,000 molecules a second.
Antibodies? What are those?
They're small proteins about 150 kilodaltons large, or about 1,300 amino acids large. They're made of 4 polypeptide chains, 2 light chains, and 2 heavy chains. The antibody forms a Y shape, and the arms of the Y are attached like hinges, making them extra flexible. The stem of the Y, Fc, is the constant fragment, and it is conserved between all antibodies of the same type. The arms, Fab, are variable regions, and they are the antigen binding fragments. The heavy and light chains in each arm can also be split into constituents. The upper parts are called the variable heavy chain, VH, and the variable light chain, VL, whilst the lower parts constituents are called the constant heavy chain, CH1, and the constant light chain, CL. The binding sites on both arms of each antibody are identical. In fact, all of my antibodies have identical binding sites. They're bob-shaped, same as my hat. Each B cell produces a specific antibody.
Binding? What is that?
It's when an antibody attaches to a unique antigen on another surface.
Bob, you really are awful at drawing, you know.
Each antibody can bind to 2 antigens, and so they can immobilize invading cells, as well as attracting other parts of the immune system.
But how do they bind?
Well, well, I don't know. I'm just a B cell. Who could possibly know that? Do you know anyone?
Ready to explore? Come on! Vamos arriba! All right! Dora, Dora, the explorer! Dora! Roots and super cool exploradora! Need your help to grab your backpack! Let's go! Jump in! Vámonos! You can lead the way! Hey! Hey! Dora! It's Dora! It's Dora! It's Dora! Swimming! Oh, man! Dora the explorer!
Hey Bob, it's me, Dora! And I'm Boots.
Hi Dora, hi Boots.
You look troubled, Bob. What's the matter?
I'm lost. I have an antibody here, but I don't know how it binds.
Don't worry, Bob. We'll help you find out more about antibody-antigen binding. Will you help us find out more about antibody-antigen binding? Great!
Oh, thank you. Gracias.
De nada.
So, this is the antibody.
Wow, that is huge. But Dora, how can we find out more about how it binds? Let's look at the variable region of Bob's antigen. Can you see the variable region? That's right. There it is. In the variable region, every chain exhibits the characteristic immunoglobulin fold. In the variable region, every chain exhibits the characteristic immunoglobulin fold. It consists of a hydrophobic core with one beta sheet on one side and one beta sheet on the other side. A beta sandwich framework. Sandwich! Oh, Boots, these sandwiches are way too small for you to eat. The loops that join the beta strands vary strongly between different antibodies. They are so variable that they are called the hypervariable loops, or complementarity determining regions, or in short, CDRs. Do you see the CDRs? That's right, there they are. CDRs are found on both heavy and light chain. Count with me how many there are. 1, 2, 3, 4, 5, 6. 6 CDRs. The light and heavy chain are folded so that all the CDRs are brought together, and this forms the antigen binding site.
The binding sites!
That's right, the binding site. The size and shape of the 6 loops, as well as its sequence combination, determine which antigen can be bound. To be precise, not all residues of the hypervariable loops are part of the binding site, but many are. For small molecules, the binding site is small. Only around 15 residues form the binding site, and some of the loops do not contribute. For larger molecules, such as proteins, the binding site is rather flat, and more than 100 residues spread over all 6 loops can take part in the binding.
But Bob, if your binding site is so perfect, can't you only catch one thing? What if there are 2 things attacking?
Well, there's more than one Bob, you know. We're all made in the bone marrow and each has his own unique antibodies, so they can attack more than one antigen.
But Bob, that doesn't make any sense. Shouldn't all B cells be identical if they're made from our DNA? Shouldn't they all produce the same antibodies?
A.
Antibody variation is first produced via VDJ recombination. B cells choose a combination of V, D, and J gene segments to produce heavy and light chains. One V and J segment together with a constant domain gene form a light chain, whilst one V, D, and J segment together with a constant domain gene form a heavy chain. In humans, VDJ recombination can generate 10 to the 6th to 10 to the 7th antibody variants. But upon further exposure to antigen, antibody affinity can increase via somatic hypermutation. Here we see 2 aligned structures of the same antibody variable region in white binding to a cellular protein in blue. The green residues in the antibody have been mutated to the red residues via the process of somatic hypermutation. Such mutations occur in antibody genes, mainly within the variable regions and hypervariable loops. Favorable electrostatic interactions and antibody loop arrangements can lead to increased affinity for the antigen. Only B cells expressing high affinity antibodies are selected. Thus, a wide range of highly specific antibodies are generated.